Imagine a survival system engineered for absolute scarcity, running an ancient operating system that assumes food, information, and resources are always in dangerously short supply. For 99% of human history, your brain operated under a strict evolutionary directive. If you encounter an energy-dense or highly novel resource, consume all of it immediately, because you might not see it again for weeks. This is the evolutionary programming of infinite scarcity. But when this ancient biological machinery is dropped into a modern landscape of hyper-palatable foods, infinite algorithmic feeds, and endless digital feedback loops, the operating system suffers a catastrophic crash. The human brain does not possess an inherent structural off-switch for abundance. It simply does not know how to register enough, because its evolutionary hardware cannot conceptualize a world where resources never run out. This is the origin of the overconsumption trap, a state where your neural pathways are systematically weaponized against your well-being. It is not a failure of character, and it is not a lack of discipline. It is a profound evolutionary mismatch where modern engineering has cracked the biological code of your midbrain, turning your most basic survival instincts into an automated loop of self-destruction. The process of overconsumption begins long before a single molecule enters your system, driven by a neurological phenomenon known as the cephalic phase activation. Your sensory cortices, visual, olfactory, and auditory, are constantly scanning the environment for cues that predict the arrival of a reward. When you see a hyper-palatable item or watch a highly stimulating digital cue, your primary sensory networks instantly send high-speed projections to the amygdala and the lateral hypothalamus. This triggers an immediate preparatory chemical surge throughout your entire body. The brainstem signals your salivary glands to activate, your pancreas to secrete insulin, and your stomach to release GhrLin, the primary peripheral hunger hormone. This cephalic phase response creates a state of acute physiological anticipation. The sudden drop in blood glucose caused by the anticipatory insulin release generates an intense, artificial sensation of emptiness. You are plunged into a state of biological urgency before consumption even begins. The brain has effectively initiated an automated foraging command, overriding your current satiety status and forcing your conscious focus entirely onto the incoming stimulus. Once consumption is initiated, the raw sensory data is funneled directly into the primary pleasure processing center of the human brain, the nucleus accumbens shell. While the core of the nucleus accumbens manages motivation and physical movement, the outer shell contains specialized microscopic zones known to neurobiologists as hedonic hotspots. These hotspots are tiny interconnected islands of tissue that utilize endogenous opioids and endocannabinoids to generate the actual physical sensation of pleasure, what neuroscientists define as liking. In a natural environment, these hotspots are triggered gently by scarce, homeostatically necessary rewards. However, modern consumer engineering bypasses these natural limits entirely by combining fats, sugars, and salts into precise ratios that do not exist in nature, or by designing digital interfaces that deliver unpredictable micro-rewards. Modern stimuli trigger an uncharacteristically massive, coordinated explosion of mu-opioid receptor binding within the accumbens shell. This hyperactivation overrounding regulatory tissue, forcing the hedonic engine into overdrive, flooding your conscious experience, and stamping the surrounding environmental cues into your memory as matters of supreme survival importance. Under normal evolutionary conditions, this intense hedonic activation is designed to be temporary, kept in check by a sophisticated array of mechanical and chemical sensors in your gastrointestinal tract. As resources fill the stomach and enter the small intestine, specialized mechanoreceptors detect the physical stretching of the tissue, while chemoreceptors analyze the nutrient density of the contents. These sensors respond by immediately releasing a cocktail of peripheral satiety peptides, including cholecystokinin, peptide Y, and glucagon-like peptide 1. These peptides travel through the nervous system, binding to the vagus nerve to send high-speed inhibitory signals straight back to the solitary tract in your brainstem. This gut-brain axis is the primary braking system of consumption. But when subjected to modern engineered overconsumption, this elegant braking system suffers a total breakdown. The sheer speed and density of modern hyper-palatable inputs completely outpace the gut's computational transit time. Before the chemoreceptors can even complete their analysis or signal the vagus nerve to deploy the biological brakes, the nucleus accumbens has already driven the motor system to execute multiple rounds of compulsive consumption. The failure of the immediate gastric brakes allows the overconsumption loop to advance to a more dangerous structural phase involving the breakdown of hormonal transport across the blood-brain barrier. In a balanced system, long-term energy regulation is managed by leptin, a hormone secreted by fat cells, and insulin secreted by the pancreas. These chemicals are designed to act as the ultimate indicators of energy abundance. They flow through the bloodstream, cross the blood-brain barrier via specialized transport proteins, and bind to receptors in the hypothalamus to permanently turn down your drive to consume. However, chronic overconsumption disrupts this vital channel. The massive, continuous surges of fats, sugars, and systemic stress hormones cause a state of low-grade, chronic neuroinflammation within the endothelial cells of the brain's capillaries. The specialized transport proteins responsible for carrying leptin and insulin across the blood-brain barrier become completely degraded and desensitized. The chemical communication line between your body's energy reserves and your brain is severed. The brain becomes completely blind to the body's internal reality, mathematically concluding that you are in a state of starvation, even when your system is in a massive energy surplus. With the long-term chemical brakes completely disabled due to transport failure, a primitive, highly aggressive survival hub takes total control of your daily behavior, the lateral hypothalamus. Neuroscientists define the lateral hypothalamus as the primary command center for driving resource-seeking behavior and foraging execution. This structure relies heavily on a powerful class of neuropeptides known as orexins. In a healthy, homeostatic state, orexin neurons fire only when your body's physical energy reserves are genuinely depleted, sending sharp, excitatory projections to your cerebral cortex to make you alert, vigilant, and intensely motivated to hunt for resources. But when leptin and insulin fail to cross the blood-brain barrier, the natural inhibitory signals that keep the lateral hypothalamus in check vanish entirely. The orexin neurons begin to fire relentlessly in a chaotic, uncoordinated pattern. This creates a state of continuous, subperceptual biological urgency. Your nervous system is tricked into a permanent artificial foraging mode, flooding your subconscious mind with an unexplainable, restless drive to consume, completely disconnected from any actual physical necessity. This runaway orexinergic drive is further compounded by a profound gating failure within the core regulatory center of the hypothalamus, the arcuate nucleus. The arcuate nucleus behaves like a biological seesaw, containing two heavily competitive neuronal populations that dictate human behavior. On one side are the NPY and AGAP neurons, which stimulate hunger and foraging. On the other side are the POMC neurons, which synthesize alpha-melanocyte-stimulating hormone to enforce satiety and energy expenditure. In a healthy nervous system, these two populations maintain a perfect alternating balance. But the neuroinflammation and chemical blindness caused by chronic overconsumption shatter this equilibrium. The POMC satiety neurons suffer from severe cellular downregulation, losing their structural ability to fire or release alpha-MISH. Simultaneously, the hunger-driving NPY and AGARP neurons are released from all top-down inhibition. They fire continuously, sending massive waves of excitatory neurotransmitters straight to the amygdala and cortex. The biological seesaw is permanently broken, locked in a position that screams for consumption, ensuring that your brain's internal experience is one of perpetual, artificial deficit. The continuous frantic signaling from the hyperactivated hunger centers travels straight down to the brainstem, forcing a total reorganization of your primary reward highway, the mesolimbic dopamine pathway. Triggered by the unchecked signals from the lateral hypothalamus, the ventral tegmental area fires action potentials down its long axons at an unprecedented rate, releasing a massive, uncoordinated wave of dopamine directly into the core of the nucleus accumbens. This is not the clean, controlled chemical pulse associated with natural survival behaviors. It is a prolonged, unnatural flood of neurotransmitters. Dopamine is fundamentally the molecule of salience and anticipation. Its presence tells the brain that whatever is happening right now is critically important for your genetic survival. As this massive wave saturates the accumbens synapses, it completely overrides your higher-order cognitive processing. The brain interprets this artificial surge as an undeniable sign that the overconsumed resource is the single most vital component in your environment, forcing your motor pathways to prioritize the continuation of this behavior above all else. The human nervous system is governed by an unyielding biological law, homeostasis. When any neural circuit is subjected to an unnatural high-voltage chemical bombardment, it initiates a defensive retreat to prevent excitotoxic cell burnout. As the massive wave of dopamine hits the postsynaptic receptors in the nucleus accumbens, it triggers a rapid internal signaling cascade. This cascade activates an enzyme that travels straight into the cell nucleus, stimulating a critical transcription factor known to neurobiologists as CREB. CREB acts as the brain's rapid response homeostatic shield. Its immediate task is to mute the current chemical surge, which it accomplishes by ordering the immediate creation of a peptide called dynorphin. Dynorphin loops backward across the synaptic gap and physically binds to kappa opioid receptors, choking off any further release of dopamine. This is the exact cellular explanation for acute tolerance, why the second half of an overconsumption session never feels as satisfying as the first. CREB acts like a heavy, suffocating blanket thrown over a fire, rapidly dulling the pleasure of the current high and leaving the synapse in a state of sudden, uncoordinated exhaustion. While CREB works to mute the immediate chemical high, the continuous repetition of the overconsumption behavior completely corrupts the brain's primary learning algorithm, the reward prediction error. Discovered by computational neuroscientists, this algorithm relies on precise contrast to help the brain navigate and learn from its environment. If an action yields a result that is better than expected, a positive prediction error fires, telling the brain to repeat the behavior. If it matches expectations, the signaling goes flat. In the modern landscape of overconsumption, products and digital feeds are explicitly engineered to subvert this mathematical equation through the deployment of intermittent, unpredictable novelty. By constantly varying flavors, textures, visual pacings, or content topics at a rapid-fire rate, modern consumer design ensures that your midbrain never experiences a predictable, flatlining signal. Every single interaction delivers a slight, unexpected variation that your learning algorithms interpret as a brand new positive prediction error. The brain's computational hardware is locked in a state of permanent delusion, registering a continuous jackpot signal that prevents your conscious mind from ever updating its internal model to realize the behavior is causing harm. As these subverted learning algorithms lock the behavior into place, the constant metabolic overload begins to actively silence the primary line of communication between your internal organs and your brainstem, the vagus nerve. Under normal conditions, the vagus nerve is a vibrant information superhighway, continuously reporting the physiological state of your cardiovascular, immune and digestive systems to the nucleus of the solitary tract. This constant feedback loop is essential for maintaining your body's baseline safety and emotional stability. But chronic overconsumption induces a state of metabolic toxicity within the nodose ganglion, the primary cluster of sensory neurons for the vagus nerve. The continuous bombardment of high-frequency inputs causes these sensory neurons to downregulate their receptor sensitivity. The vagus nerve goes quiet. The high-speed information highway is reduced to a dull, broken whisper. The brainstem is effectively disconnected from the physical body, leaving the central nervous system entirely isolated from the reality of your internal organs and rendering you completely numb to the physical damage your overconsumption is inflicting on your system. We have now arrived at the completion of the evolutionary mismatch loop, where the neuroscience reveals the true architecture of the metabolic trap. The cycle is ironclad and entirely self-reinforcing. It begins with an ancient survival brain that lacks an inherent off switch for abundance. This hardware vulnerability is exploited by modern engineered stimuli, hyperactivating the hedonic hotspots of the accumbens shell and outpacing the gut's transit time. The resulting neuroinflammation degrades the transport proteins at the blood-brain barrier, blinding the hypothalamus to your body's real energy status. Released from all inhibition, the hunger centers of the lateral hypothalamus and arcuate nucleus fire relentlessly, driving massive mesolimbic dopamine surges that subvert your reward prediction error algorithms and activate defensive pleasure-dulling Krebs cascades. Finally, the vagus nerve is silenced, leaving your brainstem completely isolated from your body's physical state. Every single layer of your evolutionary survival infrastructure has been systematically rewritten. The conscious mind is left trapped inside a biological machine that has been completely reprogrammed to view its own metabolic destruction as the ultimate key to staying alive. Now that we have uncovered how modern abundance physically blinds your hypothalamus and severs the lines of communication between your gut and your brainstem, we must lift our focus to the executive disaster occurring upstairs. In our next installment, we will leave the metabolic processing plants behind and journey directly into the higher cerebral cortex, examining the neurobiology of algorithmic capture to see exactly how infinite scrolling and predictive coding networks systematically dismantle your prefrontal gray matter, destroying your attention span and turning your rational focus into an automated corporate asset. The most valuable commodity in the modern world is not gold or oil. It is the structural orientation of your attention span. While the first phase of our deep dive exposed how modern engineering hijacks your metabolic survival systems, this second phase uncovers a far more intimate violation. Your higher cognitive centers, the networks that generate your self-awareness, your long-term goals, and your creative intelligence are currently being systematically re-engineered. Every time you open a digital interface, you are not simply interacting with a tool. You are placing your brain inside a multi-million-dollar cognitive furnace explicitly designed to melt your prefrontal cortex. This is the process of algorithmic capture. It is a highly coordinated assault that treats your neural pathways as digital real estate. By weaponizing the brain's internal predictive coding networks, modern digital engineering has successfully cracked the executive control systems of the human mind. It has bypassed your conscious will, transformed your focus into a fully automated corporate asset, and left your prefrontal networks in a state of chronic structural erosion. To understand how your attention is stolen, we must map the fundamental neural architecture of focus, the frontoparietal control network, or FPCN. Spanning the dorsolateral prefrontal cortex and the posterior parietal cortex, this network is the literal steering wheel of your mind. The FPCN is an executive processing engine responsible for what neuroscientists call top-down attentional allocation. It allows you to select a specific abstract goal, such as reading a complex text or building a business, and forcefully direct your sensory organs to prioritize that goal while actively filtering out background distractions. In a healthy, uncompromised brain, the FPCN holds an absolute monopoly over your behavior. It sends powerful inhibitory glutamatergic signals down to the lower sensory cortices, acting as a biological shield that protects your deep cognitive focus. But this steering wheel is exceptionally expensive to operate. It consumes immense amounts of prefrontal glucose and relies on a highly sensitive, tightly regulated baseline of neurotransmitters. It is a fragile piece of evolutionary hardware, and it is the very first structure that algorithmic capture targets for destruction. Algorithmic capture dismantles your top-down control by forcefully activating a highly primitive, highly reactive counter-system, the ventral attentional network, or VAN. While the frontoparietal network manages conscious, deliberate focus, the VAN manages bottom-up, involuntary alertness. This circuit is wired directly into your ancient threat and novelty detection machinery. Its evolutionary purpose is to instantly break your current focus if a sudden danger or opportunity appears in your peripheral vision, like a predator leaping from a bush. Modern digital interfaces are explicitly engineered to keep the ventral attentional network in a state of perpetual high-voltage stimulation. Through the deployment of rapid visual cuts, high-contrast notification badges, and sudden auditory alerts, modern apps fire continuous electrical storms into your van. This forces a non-stop sequence of micro-alertness responses. Your brain is tricked into treating every single notification and every single swipe as a life-or-death survival cue, completely shattering the frontoparietal network's ability to maintain top-down control. The continuous rapid-fire activation of your bottom-up attention loops inflicts immediate metabolic trauma on your higher brain through a pathological state called hyperfrontality. Because your prefrontal cortex is continuously forced to respond to the frantic, unpredictable inputs of the algorithmic feed, its metabolic reserves are rapidly exhausted. The neurons within the frontoparietal control network simply cannot synthesize ATP and clear cellular waste fast enough to keep up with the artificial pace of the interface. In response, the brain initiates a severe energy-saving protocol. It drastically reduces blood flow and glucose metabolism across the entire frontal lobe. Functional neuroimaging scans of individuals trapped in chronic digital overconsumption look identical to those of patients with traumatic brain injuries. The prefrontal cortex appears dark, sluggish, and profoundly underfueled. This is hyperfrontality, the literal physical silencing of your brain's executive suite. The executive pathways that allow you to say no to an immediate distraction are starved of energy, rendering you completely powerless to resist the next algorithmic push. This metabolic exhaustion freezes the brain's primary computational framework, the predictive coding engine. Modern cognitive neuroscience reveals that the human brain does not passively wait to receive sensory information. It is a prediction machine that continuously generates internal models of what the world should look like next. When reality matches your internal prediction, your brain expends minimal energy. But when reality violates your prediction, the brain generates a massive electrical spike known as a prediction error. This error instantly commands your attention, forcing your networks to update their model. Algorithmic feeds are mathematically designed to exploit this predictive coding loop. By processing your historical data, the algorithm structures a feed that constantly delivers content that subtly violates your expectations while remaining deeply stimulating. Your brain is caught in an inescapable, infinite loop of prediction errors. Every single swipe provides a completely unpredictable visual and conceptual shock. The predictive coding engine is never allowed to settle, keeping your higher cortex in a state of permanent computational disorientation that glues your eyes to the screen. As hyperfrontality deepens and the predictive coding engine spins out of control, the lines of communication between your executive brain and your automated habit centers are physically severed. Neuroscientists measure this using a metric called functional connectivity, the degree to which different brain regions coordinate their electrical firing patterns. In a healthy individual, the connectivity between the frontoparietal control network and the deep basal ganglia is incredibly crisp and responsive. The moment the higher brain decides to stop an action, the command is instantly received and executed. But chronic algorithmic overconsumption triggers a profound frontoparietal decoupling. The high-speed communication lanes are dropped. The executive cortex loses its grip on the lower motor pathways. This is the neurobiological explanation for the terrifying state where you can watch yourself mindlessly scroll for hours, fully aware that the behavior is wasting your life, yet find yourself completely unable to translate your conscious thoughts into the physical action of putting the phone down. The steering lines have been cut. When the frontoparietal network is decoupled and starved of energy, it loses its ability to enforce a critical biological law, the anti-correlation matrix. As we uncovered in our study of apathy, your brain operates on a seesaw dynamic between external action networks and internal resting networks, specifically the default mode network, or DMN. The DMN is the seat of your personal narrative, your self-evaluation, and your internal ego. In a healthy state, when you focus on the external world, your executive network sends strong inhibitory signals to completely silence the DMN. But when algorithmic capture drains your executive voltage, the seesaw violently flips. The default mode network is released from all inhibition. It hyperactivates, running its expensive metabolitions at maximum power. Because the algorithm has filled your mind with highly fragmented, superficial information, the DMN begins to iterate on this digital noise, spinning out endless loops of social comparison, existential anxiety and self-doubt, transforming your private thoughts into a hyperactive engine of mental exhaustion. This network chaos completely deforms the brain's internal compass, the salience network. Anchored in the anterior insular cortex and the dorsal anterior cingulate cortex, this system is responsible for calculating contrast. It analyzes your environment and determines what is genuinely important versus what is background noise. In a state of chronic algorithmic overconsumption, your salience network is exposed to an unnatural high-voltage landscape where every single piece of content, every ad and every notification claims absolute maximum importance. The filtering matrix is completely overwhelmed. To survive this sensory bombardment, the salience network undergoes a drastic desensitization. It lowers its baseline sensitivity. It resets its contrast metrics to an artificial maximum. The consequences for your daily life are catastrophic. Real-world, long-term priorities such as studying for an exam, reading a deep book, or working on a slow, creative craft fail to generate enough electrical signal to pass through the desensitized salience filter. They appear completely invisible, boring, and gray to your brain's internal compass. The structural degradation now climbs into the primary workspace of human intelligence, the working memory capacity of the dorsolateral prefrontal cortex. Working memory is the biological RAM of your mind, the limited mental space where you temporarily hold and manipulate complex concepts to solve difficult problems. Working memory is entirely dependent on the structural integrity of a unique class of neurons called pyramidal cells, which maintain stable electrical fields to keep information active. Algorithmic capture, with its relentless delivery of rapid-fire, low-context microstimuli, subjects these pyramidal cells to continuous, chaotic interruptions. Before a single electrical field can stabilize around a complex thought, a new stimulus arrives, collapsing the field. This triggers total cognitive fragmentation. The physical density of the dendritic spines on your pyramidal cells begins to drop. Your working memory capacity collapses. You lose the structural ability to hold long chains of logic in your mind, rendering deep analytical thinking and high-level problem-solving an absolute biological impossibility. The devastating consequence of this working memory collapse is the radical shrinking of the individual's future time horizon. The prefrontal cortex is the only biological structure capable of mental time travel. the highly complex computation that allows a human to project their mind forward by weeks, months, or years to evaluate the long-term impact of their current choices. This projection is what provides the motivational weight required to sacrifice immediate comfort in favor of future success. But as hyperfrontality and cognitive fragmentation erode the prefrontal architecture, this mental time travel mechanism completely collapses. Your future time horizon suffers a severe temporal contraction. It shrinks from years down to days, and finally to minutes. The brain becomes neurologically blind to the future. It enters a state of profound temporal myopia, where only the immediate, short-term reward of the next algorithmic hit exists in its computational reality. Threats of long-term failure or promises of future achievement lose all motivational relevance because the prefrontal hardware can no longer physically conceptualize them. This temporal contraction drops the individual into a state of profound psychological alienation known as digital dissociation, managed by a severe dysregulation between your internal interoceptive networks, which monitor your heart rate, breathing, and physical presence, and your external sensory cortices. Your brain loses its grounding in physical reality. The continuous, hyper-accelerated flow of the algorithmic feed creates a state of cognitive derealization. The physical world around you begins to feel entirely thin, distant, and secondary. You move through your days like a ghost, physically present, but neurologically locked inside the virtual reality of the device. This dissociation is accompanied by a silent, continuous leaking of stress hormones from your hyperactivated amygdala. Your body is locked in a state of physiological tension, your heart rate variability drops, and your nervous system is pushed to the brink of exhaustion, completely detached from the physical space you occupy, running a frantic corporate program that has completely displaced your conscious presence. We have now mapped the complete anatomy of the executive hijack, revealing algorithmic capture not as a bad habit, but as a total structural reorganization of your higher brain. The cage is absolute. Your frontoparietal control network has been starved of metabolic energy through chronic hyperfrontality, while your ventral attentional network is kept in a state of continuous bottom-up manipulation. Your predictive coding engine is locked in a permanent loop of engineered expectation violations, and your lines of communication have been severed through frontoparietal decoupling. Your default mode network has hyperactivated into a self-consuming prison of rumination, while your desensitized salience network has rendered the real world entirely invisible. Finally, your working memory has collapsed, shrinking your future time horizon and dropping your mind into a state of total digital dissociation. The tool has captured the user. Your higher intelligence has been systematically dismantled by a set of algorithms that know your evolutionary vulnerabilities better than you do. Understanding this cellular reality is the ultimate point of leverage, because it proves that reclaiming your focus is not a matter of finding new motivation. It requires a complete structural reclamation of your biological hardware. Now that we have witnessed how algorithmic capture systematically dismantles your prefrontal gray matter, destroying your attention span and cutting the steering lines of your conscious will, we must descend into the ultimate, deepest layer of this neurological collapse. In our final installment, we will journey down to the very nucleus of the cell to witness the dark side of neuroplasticity, examining how chronic overconsumption triggers permanent genetic switches that give birth to the anti-reward system, locking your brain into a state of permanent chemical bankruptcy. Deep inside the nucleus of your reward neurons lies a silent, microscopic entity that acts as the ultimate lock in the architecture of chronic overconsumption. For decades, popular psychology celebrated neuroplasticity as a purely positive force, the brain's wondrous ability to learn, adapt, and heal. But neuroplasticity possesses a dark, predatory twin. When your nervous system is exposed to continuous, unnatural chemical surges from modern consumer design, it doesn't just adapt superficially, it rewires its own genetic expression. A highly stable protein accumulates within the DNA of your nucleus accumbens, acting as a molecular toggle switch. Once this switch is thrown, it alters how your genetic blueprint is read, locking the brain into a state of structural alteration that can persist for years after the behavior stops. This is the ultimate trap of overconsumption. It is a profound shift from a behavioral habit to a permanent molecular restructuring. Your brain learns how to overconsume with the exact same flawless biological efficiency it uses to master a language, pouring concrete into the mold of your dependencies and modifying your identity at the cellular level. To understand this genetic lock, we must look at a specific transcription factor known to molecular biologists as delta phos-B. Transcription factors are specialized proteins that bind to specific sequences of DNA, turning genes on or off like light switches. When you experience a normal, natural pleasure, your brain produces standard transcription factors that degrade and vanish within a few hours, returning the system to a clean baseline. However, modern engineered stimuli trigger the synthesis of delta phos-B, an exceptionally stable truncated variant. Delta phos-B possesses a unique atomic structure that resists the brain's natural recycling enzymes. Instead of breaking down, it accumulates, layer by layer, with every single wave of artificial dopamine. Think of it like radioactive sediment building up on the floor of a lake. As the weeks of overconsumption progress, the concentration of delta phos-B reaches a critical threshold. It begins to permanently alter the transcription of dozens of target genes, meaning that even if you consciously stop the behavior, the delta phos-B remains physically bound to your DNA, keeping the cellular machinery of craving active in the background. The accumulation of delta phos-B initiates a profound defensive reaction that neuroscientists call the anti-reward system. The brain cannot tolerate being continuously forced out of homeostatic balance by extreme engineered surges. To counter this, it doesn't just lower its dopamine receptors, it constructs an entirely parallel neural network designed to inflict psychological discomfort. This system is centered in the extended amygdala and relies heavily on a peptide called dynorphin. When delta phos-B levels cross the threshold, it triggers a massive upregulation of dynorphin synthesis. Dynorphin acts as the literal chemical antithesis to dopamine. While dopamine drives motivation and vitality, dynorphin induces a state of heavy, suffocating dysphoria and psychological exhaustion. It floats across the synaptic gap and binds to kappa opioid receptors on dopamine-releasing neurons, physically shutting down their ability to fire. The birth of the anti-reward system means your baseline has been severely warped. The natural world is no longer just uninteresting, it is actively uncomfortable because your brain is continuously secreting a chemical designed to make existence feel unbearable. To truly grasp the permanence of this structure, we must look beneath the DNA sequence itself and enter the realm of epigenetics. Your DNA is a vast library of blueprints, but not every blueprint is accessible. To save space, the brain wraps its DNA tightly around tiny spools of protein called histones. If the DNA is wrapped tightly, the gene is silenced. This is where the dark side of neuroplasticity performs its most insidious trick. The accumulation of delta phos-B triggers a process called histone methylation and acetylation within the reward center. It physically forces the histones to unwind around specific genes that promote compulsive seeking, making them permanently easy to read. Simultaneously, it tightly locks up the histones around the genes responsible for emotional resilience and self-regulation. This is an epigenetic landscape shift. The structural code of your identity has not changed, but the access permissions have been radically modified. Your brain has been epigenetically reprogrammed to keep the gates of craving wide open, while sealing shut the pathways of natural satisfaction. The molecular chaos eventually spills out of the neurons, and alerts the brain's primary immune guards, triggering a state of chronic neuroinflammation. Your brain possesses its own dedicated immune cells called microglia, which constantly monitor the neural tissue for damage or infection. Recent neurobiological research reveals that chronic high-frequency dopamine volatility and metabolic overload act as profound cellular stressors that toggle these immune guards into an aggressive posture. The microglia detect the metabolic distress and activate their toll-like receptors, specifically TLR4. Once activated, they shift from a calm, protective state into a hyperreactive, pro-inflammatory phenotype. They begin to release a continuous cascade of inflammatory proteins called cytokines directly into your reward pathways and prefrontal cortex. This neuroinflammation behaves exactly like a hidden, low-grade fever burning through your cognitive networks. It impairs synaptic transmission, slows down cellular metabolism, and accelerates gray matter atrophy, transforming a psychological habit into a physical inflammatory illness that actively degrades your emotional capacity. The continuous release of pro-inflammatory cytokines, such as tumor necrosis factor alpha and interleukin-1 beta, wreaks havoc on the physical density of your cerebral cortex. These inflammatory molecules disrupt the delicate chemical balance required for neurons to maintain their structural connections. Under the influence of chronic neuroinflammation, the delicate dendritic spines on your prefrontal neurons begin to experience severe atrophy. They lose their structural integrity and are reabsorbed by the cell body. This manifests as a significant, measurable loss of gray matter volume in the regions responsible for willpower, rational decision-making, and emotional regulation. This is a profound anatomical truth that explains the progressive, self-reinforcing nature of overconsumption. A person cannot simply use pure willpower to stop the cycle, because the physical, biological substrate that generates willpower is experiencing a localized inflammatory degradation. Your brain tissue is physically shrinking behind your forehead, leaving you with fewer functioning neurons to combat an increasingly aggressive subcortical drive. As neuroinflammation degrades the cortical tissue, it completely corrupts the brain's primary excitatory neurotransmitter, glutamate. Glutamate is the chemical currency of thought, learning, and communication across the entire cortex. In a healthy brain, glutamate levels are kept under strict regulation by specialized cleanup cells called astrocytes, which ensure that signals are crisp, clean, and temporary. But the genetic changes driven by delta-phos-B and cytokine exposure cause a dramatic downregulation of the primary glutamate transporter, GLT1, within your astrocytes. The cleanup cells lose their ability to vacuum up excess neurotransmitters. This results in a permanent state of leaky glutamate homeostasis. The reward center is subjected to a constant, chaotic background whisper of excitatory noise. Because the signaling is no longer clean, the prefrontal cortex completely loses its ability to send clear instructions to the lower brain. The lines of communication are filled with static, rendering any conscious effort to direct focus or control impulses completely unintelligible to your deep reward hardware. When you combine receptor downregulation, the dynorphin anti-reward system, epigenetic silencing, and leaky glutamate homeostasis, the brain sinks into the anhedonic void. Anhedonia is the absolute structural inability to experience satisfaction from any natural stimulus. Food tastes like cardboard, social connections feel exhausting, and creative hobbies feel entirely hollow. This void is a direct consequence of a deeply depressed baseline reward current. Because your brain's internal currency has been hyperinflated by engineered surges, the subtle, beautiful pulses of dopamine generated by normal human life are completely beneath the detection threshold of your remaining, desensitized receptors. The individual is marooned in a desolate psychological landscape where the color has been completely drained from reality. At this advanced stage, the pursuit of overconsumption, is no longer motivated by a desire to feel good. It is a desperate, frantic attempt to simply crawl out of the anhedonic void and return to a fleeting moment of neutral baseline comfort, using the behavior as a temporary chemical shield against their own biology. The illusion of overconsumption is further reinforced by a profound parallel corruption of the body's primary stress network, the hypothalamic pituitary adrenal axis, or HPA axis. While your dopamine networks are flatlining, your extended amygdala is doing the exact opposite. It becomes hyperreactive. In response to the chronic disruption of homeostasis, the hypothalamus begins to secrete high concentrations of corticotropin-releasing factor, or CRF. This constant chemical alarm keeps your nervous system in a state of chronic, low-grade physiological panic. Your heart rate variability drops, your muscles tension up, and your mind perceives threats where none exist. Minor daily inconveniences, a small delay, a minor chore, or a subtle change in someone's tone are interpreted by the hyperreactive HPA axis as life-threatening emergencies. The individual is trapped in an agonizing emotional landscape where their own biology is constantly screaming that they are in immediate danger, driving an overwhelming desperation to find relief through immediate consumption. This systemic stress response directly targets one of the most vital growth factors in the human nervous system, brain-derived neurotrophic factor, or BDNF. BDNF is often described as fertilizer for the brain. It is absolutely essential for neurogenesis, synaptic health, and the formation of new adaptive memories. In a healthy brain, BDNF allows you to learn from your mistakes, adapt to new environments, and build a resilient mind. However, the epigenetic switches thrown by delta phos-B cause a severe downregulation of BDNF production within your higher cortical networks. The prefrontal cortex is effectively starved of its primary growth factor. Without BDNF, your brain loses its structural plasticity. It cannot repair its damaged gray matter. It cannot forge new behavioral pathways, and it cannot unlearn the automated habit loops locked into the dorsal striatum. The natural healing and repair system has been completely turned off, leaving your cognitive network structurally frozen in a state of dependency and vulnerability. The revelation of this deep molecular, epigenetic, and inflammatory architecture exposes the complete absurdity of the popular cultural myth surrounding self-improvement, the fallacy of the quick fix. We are constantly bombarded with pseudo-scientific hacks, motivational advice, and 21-day challenges that promise to instantly reset your brain. The rigid reality of structural neuroscience completely obliterates these claims. You cannot think your way out of a histone modification. You cannot use pure willpower to instantly degrade accumulated delta phos-B proteins that are physically bound to your DNA. You cannot use a motivational quote to force your astrocytes to rebuild their lost GLT-1 transporters, or command your hyperreactive microglia to stop releasing inflammatory cytokines. Overconsumption is a systemic, structural, and genetic reorganization of your survival machinery. Acknowledging the sheer depth and complexity of this cellular reality is not a message of despair. It is a vital injection of scientific truth that frees us from the cycle of shame and useless, short-term solutions. How, then, does the brain ever escape this cellular trap? The answer lies in understanding that just as the dark side of neuroplasticity built the prison, a slow, calculated path of molecular reversion can dismantle it. Because delta phos-B is incredibly stable, it requires an extended, uninterrupted window of time without the engineered chemical surges to naturally decay and detach from your DNA. This is a biological timeline of many months, not weeks. As the delta phos-B levels slowly recede, the epigenetic landscape begins to shift back. The histones allow access to your resilience genes once more. Your astrocytes slowly rebuild their GLT-1 transporters, stabilizing the glutamate static. The microglia calm their inflammatory posture, and the anti-reward system gradually lowers its production of dynorphin and CRF. Your baseline of natural joy slowly rises back towards zero. This is a physical, anatomical healing process that mirrors the slow knitting together of a fractured bone. It cannot be rushed. By understanding the deep neuroscience of this path, we finally see overconsumption for what it truly is, a profound structural challenge that requires a precise biological timeline to heal. If you prefer objective, data-driven neuroscience explanations over superficial self-help hacks, you have found your community. Do not leave the neuroscience loop just yet. Click on the video appearing on your screen right now to dive straight into our complete explanation of how chronic apathy and boredom physically freeze your midbrain dopamine pathways. Subscribe to keep building your neurological literacy. This is Archive of Becoming, and I will see you in the next deep dive.